Um,
Hi. Would you like to try sharing just so that we know that you can share your screen without any issues for later?
sure.
Excellent. Awesome.
Working?
Working.
Okay. So I will be right back, okay?
Okay.
Hey, Sophia, can you hear me well?
Yes, I can.
Okay. Whoa.
Ooh.
Okay. Oh,
Oh, there you are.
here. Yeah.
Well, I'm excited to meet you. I, when Erica was visiting Tucson, I was talking to her about my seminar club, and she's like, I know the scientist for you. So I'm really excited that this worked out.
Oh, thank you. Erica is so sweet.
Yeah, she's really sweet.
I hope I'm not disappointing you.
No, I'm sure you won't.
So I met your father here in Brazil.
Really?
Yeah.
What do you think of him? Is he okay?
Yes. I
Yeah.
missed his lecture,
Hmm.
but I met him just after his lecture.
Okay,
So he seems well.
cool. I like him, so
Ha-
that's just my opinion.
Have you been to the South America?
Um, I have not been to Brazil, but I have been in South and Latin America pretty extensively when I was a kid. My dad, for a semester, he taught in Argentina, so we all lived there for a semester. And he did that in Chile and Guatemala too. So I've lived in those countries as well. And I've been to um, Ecuador and Mexico, Costa Rica, Uruguay. I don't even know where else. I've been all over the place. But it was interesting because most of my experiences in Latin America were experiences where we went and we lived somewhere or it was for a research project. And so, not necessarily the typical tourist experiences that one might have.
Yeah, this is the best experience, right?
Yeah, I think
Because you
so.
can really feel the place.
Yeah. And I
Yeah.
would love to shape my career in a similar way to how my dad did so that I can also have research projects and go and travel and do that kind of thing. I think that's really cool.
Yeah. So what's your major?
So I am a PhD student in medical pharmacology, and there was a really weird thing that happened like 30 years ago at the U of A where the pharmacology professors started hating each other, and so they split into two departments of pharmacology. And one is medical pharmacology, and the other one is pharmacology and toxicology. So my major is medical pharmacology, and my minor is pharmacology and toxicology. It's just so dumb.
Oh boy. And how are you doing? Are you liking, how are you enjoying
I'm
your
really
experience,
enjoying it.
your PhD? Yeah.
Yeah, I'm really enjoying it. I very much lucked out with my advisor, my committee, and they're letting me do a kind of weird project for our department. Our department does a lot of work with opioid receptors, cannabinoid receptors, um, drug development, figuring out what orphan receptors do. And I'm doing a toxicology study, which is just totally different. Hey, Caroline. But I love it. And they've, supported me with good ideas and funding and all the things. Hey, Caroline. So yeah, I'm really grateful.
How have you been, Sophia?
I've been good. How have you been?
I've been really well. I had interviews for the ABVS program like mid-January. They went really well, and waiting to hear back in March, it seems like.
Okay, awesome. I have gotten information back about my F-31 proposal, but every time I get information, it's like, this is the next scheduled day when we will meet to discuss it. So now I know March 18th is the next time that they'll meet to discuss it. And I think that's the last time, but I don't really know.
What is there a deadline for them?
It passed.
Well, good luck, and I
Yeah.
wish you all the best. Hopefully, you
Thank
get it.
you. Thank you. I think we've just had like so much chaos with the government shutdowns and everything that they start and then they stop and they start and they stop. But hopefully I get funded. We'll see. Um,
Is the grant NIH funded?
yeah.
Uh,
And is this grant to cover your fellowship, your scholarship, or
Yeah.
your research?
It would be for my tuition.
Okay,
I'm
for
on a
tuition.
fellowship right now for my tuition, but it's typically only a 2-year fellowship, and I'm in my second year. So I'm trying to acquire funding for myself starting in June, and an F-31 would be a wonderful way to achieve that. But I applied for funding at the NIEHS, and that's not a great agency to have applied to. So we'll see.
So other options you have, you pay yourself your tuition? It's something like that, or you have to have a sponsor or something.
Worst case scenario, I'll be put on a grant for one of my
All
mentor
right.
projects. Yeah. We
Mm-hmm.
don't actually have TA positions in my major department, which is really weird. Hey, Shofu. Um, so that's just really strange, but that's the way it is. And so if I don't get a fellowship, I'll just be on a grant.
Hey. Because here in Brazil, since we get a PhD fellowship, it covers the whole PhD, which it's
That's better.
4 years. Yeah.
So I was reading a little bit about you, and I saw that you did your postdoc at Stanford and the Butantan Institute. How did that work?
Yeah. Yeah. I did 1 year at Butantan and 1.5 years at Stanford. Because I started a project when I was doing my PhD, and then I was able to follow up the same project at Stanford. We have in Brazil, we have when we are a PhD student, you can have like a sandwich scholarship, it's the name, and you do like 1 year abroad. So I did my 1 year PhD abroad, and then I met Daria from Stanford, and then I came back. I finished my thesis, and then I went back again to finish the project I started, in parallel in my PhD.
That's really cool. Caroline, that
Yeah.
sounds like your cup of tea.
That would be awesome. I really, yeah, I was hoping to go and maybe do graduate school in Columbia, but it's just you have to pay for it instead of them paying you. And then I got a really good opportunity to work with a professor here. But I would love to go somewhere else and do some studying. I'm interested in studying natural products and mostly like I want to study like plant natural products that come from places that we haven't explored, like the Amazon and South America. So one of our friends, Sophia knows him, his name's Eric. He's getting a PhD in drug discovery here at the University of Arizona, and he's from Brazil. And so, but I would love to be able to like source new plants and then characterize them and maybe see what they do. And the professor that I'm most interested in working with already kind of does that. So that would be a great segue into the field.
Okay, can you say again the name of your supervisor?
I'm not in at the university right now. I applied to a PhD program for the upcoming fall. So the last person that I worked with, they have a bachelor's degree in biochemistry from the University of Arizona, and the last PI that I worked with was Dr. Greg Thatcher, who is he is a PI in drug discovery development with Alzheimer's disease and a couple other diseases as well.
Okay.
But with NIH funding cuts, so I went and worked in industry for a little while. I went and worked at Roche Tissue Diagnostics, where they do cancer diagnostic assay development. But I found it really boring, and I didn't like it, and so I left. And so I'm in this weird interim place where I know I want to get a PhD. I know I don't want to be in industry. I know I want to be a professor. But with the funding cuts, there's no positions available really at the university here in Tucson for people with bachelor's degrees. It's like they have to pay people who are getting their degree or or people who are graduate students or people who already have PhDs to manage those people. So like it kind of feels like the positions that were available for people with bachelor's degrees in academia they've dried up for the moment. But so that's kind of where I'm at.
The program that Caroline is applying for is a really cool program that we have here where essentially you get accepted to be a PhD student and then you can actually choose between 4 or 5 different departments that all have different research topics and opportunities. And you get to rotate in any of those departments to figure out what you want to do.
Yeah, the same as in Stanford. You do a rotation and then you can pick what you like most.
All right, well, it is 4 p.m., so I guess we can get started. Thank you, everyone, for joining. Dr. Zambelli and I actually met via email in a really strange way. We were introduced by a friend who works at the same institute as her, who I was just at dinner talking about the seminar club, and she was like, I know the scientist for you. So here we are. But Dr. Zambelli actually does a lot of research that is very similar to what we are interested in at the U of A. And her research has included topics like the opioid receptors, cannabinoid receptors, obviously a lot of mitochondrial dysfunction work, but I think it's highly relevant to a lot of our interests. So I think this will be a good one. All right.
Thank you, Sophia. Thank you, everyone, to be online, giving your time to discuss a little bit of what we has been doing the past years. So, as Sophia mentioned, I'm a pain scientist, but I'm also interested in targeting mitochondria for treating chronic pain. So just a little bit of background for you. I'm sure you are aware of it. So here is the Butantan. We are, hold on, hold on. Just a little bit. I'm just trying to use the presenter view. Okay. Now, so this is Butantan Institute. So we are located in São Paulo. It's extensive. Can you see the movie? Or yeah, it's moving? Okay. It's an extensive green area of about 700,000 square meters surrounded by the science park with museums and laboratories and so on. So we are quite lucky because it's a big city, but we still have green areas and my lab is just in the middle of everything. Okay. And this is my team. So we have, like in your lab, I'm sure, master students, PhD students, and postdocs. And you are all welcome to spend a couple of time in your summer. So we are spots for summer internships and so on. So you are more than welcome. And why we are so, is there anyone here that studies pain? You have a pain scientist in your group?
A lot of the people who are here study pain. Shofu,
Oh,
Cody, I do, Caitlin does, Caroline might next year. FYI, I just want to let you know that we can see the view now where the little slides are down below. I don't know what it's called. Um, but the main slide is in the top
okay.
left corner.
This is not so good for you too.
Perfect.
What about now? Okay,
Perfect. Yeah.
that's much better. Okay, so why do we study pain? Because around 30% of the whole population feels pain, right? And in US, only in Brazil, around 85 million people has some kind of pain that's debilitating, so they have to look for a health system and so on. And this is a very cool data. I know it's from 2016, but it's still up to date because around 100 million people suffers for some kinds of pain. And pain costs more to the health services than costs the diseases that more kill, right? Like heart disease, cancer, and diabetes. So that's why there are so many groups interested in finding new molecular mechanisms and new drugs to treat pain. So, okay, I don't need to give you this brief reminding, but it's always good to remember that the pain pathways are basically made up of afferent primary nociceptive neuron that we call nociceptor, the dorsal root ganglia that we call DRGs, where are the cell bodies are located, right? They are pseudo-unipolar neurons, and the cell body is located in the DRG. Can you see my pointer? Okay, good. And important to remember that one of the afferents goes to the spinal cord, the dorsal horn spinal cord, where the pain is processed and do synapses with the secondary nociceptive neurons that goes to the supraspinal regions, especially thalamus and somatosensory cortex, where the pain is localized, right? Okay, so you probably are aware that more recently, pain has been divided in nociceptive pain, nociplastic pain, and neuropathic pain. So for the nociceptive pain, mostly is like a defense mechanisms, such as a pain induced by bone fractures, joint inflammation, muscle spasms, burns, and ulcer. And also the inflammatory pain is usually called as an nociceptive as well, and sometimes can be autoimmune or just for post-operative. So in other words, the nociceptive pain is quite of our mechanism of defense. But sometimes this nociceptive pain becomes chronic, and then we have to pay more attention to it, right? And another important kind of pain is a neuropathic pain, which is basically an injury in the peripheral or central somatosensory system induced by a trauma, neurodegenerative diseases like Parkinson's, autoimmune like multiple sclerosis, HIV, diabetes, cancer, or chemotherapy. And the nociplastic pain is when there is no evidence of tissue or the somatosensory system injury, but the pain is still there, like fibromyalgia, complex regional pain syndrome, and visceral pain syndrome. I don't know if everybody's aware of a complex regional pain syndrome, but it's very common when a person use a cast, and like you fracture your bone and you use a cast and the fracture is gone, you are all healed, but the pain is there. Probably because when you have a ischemia, and the ischemia can manage to induce chronic pain. And the injury is gone, everything is gone, but the patient still feels pain. And nowadays, there are also the mixed pain that can be a combination of all of those, and this is why it's so difficult to treat pain, right? Of course, there are opioids, cannabinoids, there are antidepressants, there are several classes of painkillers, but we still need to find new pharmacological targets, because the majority of these painkillers, they do induce gastric problems or rewards, right? And people can die from the side effects. So we urgently need new pharmacological targets to treat pain. So back in 2014, I started looking at the mitochondria, and I'm sure you are aware of it, that it's a super important organelle responsible for energetic metabolism, redox balance, ion homeostasis, and cell death. But more recently, an impairment in the functions of mitochondria has been gaining more attention because it's related to several degenerative diseases, such as obesity, cardiovascular disease, but also neurodegenerative disease, mitochondrial disease, and neuropathic pain. So in my studies, I looked at two specifically, let me just move my, okay, two specifically functions of mitochondria, which are the detoxification, mainly of oxidative stress and aldehydic detoxification. And in the second part of this talk, we'll speak a little bit about the quality control, how mitochondria control the fusion and fission process. Okay, first we look at detoxification. So how we did that. Okay, you probably know an mitochondrial enzyme called aldehyde dehydrogenase 2. It's a very well-known enzyme that is responsible for metabolizing acetaldehyde from the alcohol intake, right? But when we have an injury, like a hypoxia or even inflammation, we have ROS production, lipid peroxidation, that culminates in aldehyde productions. And what is the problem? Aldehydes are pretty more stable than ROS, and they can make adducts with important proteins in the cell, and can make impairment in the cell functions. And a couple of years ago, studies were demonstrated that there were some relationship between the accumulation of aldehydes and pain, more specifically 4-hydroxynonenal, which is a lymphatic aldehyde that is able to induce pain. We will talk a little bit more about it, but especially by activating TRPA1 receptors. You probably have a friend, or if you are Asian descent, I don't remember the pronunciation, okay? And around 40% of the East Asian population, they have this phenotype after drinking alcohol intake, which is like this red face, tachycardia, nausea, and this is called Asian flush. I'm sure you know someone that has the Asian flush, and it's caused by a mutation in the aldehyde dehydrogenase 2, where happened a switch of a glutamic acid to a lysine. And this mutation confers a reduction in around 60% of the activity in the heterozygote person, and it can reach around 100% in a homozygous. Okay, but why I'm looking at it? Is there any involvement in ALDA2 and pain? I told you that ALDA2 is metabolizing aldehydes, and aldehydes can be painful. So what happens if we are able to target this enzyme and look at pain? So we use two strategies: the loss of function and gain of function of this enzyme. So for the loss of function, we use a mutant mice with this that mimics the same Asian mutation. So they have an impairment in the ALD2 activity. So we can clearly see here the wild type mice that received ethanol, and the twister2 mice, I will call them twister2, right? Because it's the name of the mutation. The twister2 mice, they are clearly more prostrate after alcohol intake. So in fact, they have an impairment, a decrease in the ALD2 activity from the liver when compared to the wild type mice. And when we give them when we measure the acetaldehyde levels after the alcohol intake, we can see that these mice, they do have increased blood acetaldehyde levels when compared to the wild type mice. Okay, so we had a model. And what we did, we injected carrageenan in the hide paw to induce inflammatory pain, right? I can't use the word pain for mice, right, and rats, but let's assume that I'm saying nociception, but I'll call it pain, okay? Because they don't tell us. And I don't know if you are aware of the up and down technique when we apply the von Frey hair and we take the base, the nociceptive threshold. If you want to interrupt, I don't know how you manage the conversation. You can interrupt me. For me, it's fine if you have any question, okay?
Yeah, we do a few different types of von Frey. So a lot of the people here actually do hindpaw von Frey and periorbital von Frey. And in my lab, we use periorbital von Frey with the up-down method is the
Okay.
typical way of doing it.
Okay, so everybody knows what I'm talking. I will open the door for my cat. He's crazy. Pode entrar. So what we did, so we injected carrageenan and we saw a decrease in the pain threshold, right? When we compare to the baseline levels, and for our surprise, the twister2 mice, they have an increased hypernociception when compared to the mutant mice. And when we took the paw, we look at the 4-HNE levels, the formation of adducts in the hind paw, in the inflamed paw. We clearly see that the wild type mice here in red, they have increased levels of 4-HNE when compared to the wild type mice. Okay, so what happens if we give function to ALDA2? So we use the small molecule called ALDA1, which is allosteric agonist and a molecular chaperone that corrects the ALD2 structural defect of ALD2. So we can see the wild type, and here we have an impairment in the structure, which confers the mutation, which confers the decrease in the activity of ALDA2. And when we give ALDA1, it corrects the structure and it increases the activity by 2 times, twice in wild type and 11 times in the mutant enzyme. So it corrects the defect and increases the activity. And it confers some cell protections in different models by decreasing the aldehydic loads. So we tested this in our model. So for this, we gave Alda-1 15 minutes before carrageenan, and then again and again because of this short life of Alda-1. And we measure pain. So here again, so this is the baseline. Here's the vehicle for Y type, the mutant mice with increased hypernociception, and Alda-1 correcting the enzyme and increasing its activity were able to induce analgesia or to prevent the hypernociception induced by carrageenan. And when we took the paw of those rodents, we clearly see that Alda-1 prevented the aldehydric load in the inflamed paw. We also measured other aldehydes like malonaldehyde and acetaldehyde. And we found it increased in the inflamed paw and decreased when we treat them with Alda-1. Okay, so we showed that inflammation increases the aldehyde load. So what happened if we give an exogenous aldehyde in the acute pain? So there was this pain. Okay, so we sorry, I think I was a little bit confused. So we showed that endogenous production of 4-HNE, and now we asked if this enzyme was also important if we give an exogenous 4-HNE. And there was this very important paper from 2007 showing that 4-hydroxynonenal causes pain and neurogenic inflammation by activating 3PA1 receptors. And I'm sure you are aware of the 3P channels family, right? Because it gave the Nobel Prize to these two incredible scientists, right? And I'm sure you know that 3PV1 is sensitive to capsaicin, of course, to the temperature, but also to capsaicin, and 3PA1 to mustard oil, garlic, wasabi, and cannabinoids. So we checked the 3PA1 receptors. So just remember, when activate 3PA1 receptors, we have an increase of calcium influx and release of substance P and CGRP, which are important neuropeptides involved in pain. And when we inject 4-HNE in the palm, and just before we give a 3PA1 antagonist, so first of all, we see that 4-HNE, when we administer it exogenously, we do have a decrease in the pain threshold. In other words, 4-HNE induces pain in rodents. And when we pre-treated those mice with a 3PA1 antagonist, we are able to prevent the hypernociception. So suggesting that 3PA1 mediates 4-HNE effects. So we confirm the previous data from the literature showing that 4-HNE targets 3PA1 receptors and then induces pain. And what happens if we give 4-HNE to the mutant mice that don't have the enzyme that metabolize 4-HNE? So they have the same amount of pain when compared to the wild-type mice, but it's long-lasting. So suggesting that the mutant mice are not able to wash out the 4-HNE that we inject in the rind pod. Okay, so from now what we had, that the impairment in ALD2 activity increased inflammatory pain, and that activating ALD2 with Alda-1, we were able to decrease inflammatory pain. And these were all mediated by aldehydes, right? Okay, but it's quite easy to treat inflammatory pain. What happens if we move to a chronic pain? So we chose to study the neuropathic pain that, as I mentioned, it's an injury in the somatosensorial system, and it's quite common, quite prevalent in the individuals, and can be induced by trauma, neurodegeneration, autoimmune infections, metabolic, and toxic. So here I will show you two examples in autoimmune and toxic. We have studies in almost all of these kind of conditions, but I just picked something to show you today, okay? So we chose the multiple sclerosis because we were able to develop this model in our lab. So it's a quite common disease affecting mainly women, young women, and there is no cure, right? And it's an autoimmune disease. It happens after a disruption in the blood-brain barrier. It induces demyelination, axonal degeneration, impairment in neurological functions, but especially 70% of the patients have some kind of pain. And we wrote this mini review that you can check if you are interested, and showing how the mitochondrial dysfunction can contribute to multiple sclerosis. So in our group, we decided to look at the aldehydic load, and if controlling the aldehydic load levels, we were able to manage to control the multiple sclerosis. So we chose a very common model for multiple sclerosis. I'm not giving you so much details, but if you are interested, we can talk more later. I'm just giving the nicer results. And so we injected myelin oligodendrocyte glycoprotein, and we induce an autoimmune response. So these mice develop a progressive sclerosis, multiple sclerosis symptoms. They do developing before the motor deficits. So basically, they have inflammation, neurodegeneration. They have important motor deficits that we are able to quantify using scores. And we also able to measure the pain threshold before the motor symptoms. It is important because since we are doing a test that depends of the movement, right, of the rodent, so we have they have to be able to show the behavior. So what we showed that the model is an experimental autoimmune encephalomyelitis, which we can just call the multiple sclerosis model. So they have motor impairment, weight loss. They do develop hypernociception and neuroinflammation. So what we saw here are the wild type and the two star 2 mice. So, and these are the scores for the motor impairment. So we can clearly see that the two star 2 mice, in other words, the impairment in ALD2 activity increases the score of the disease. We checked the 4-HNE levels in the spinal cord, and we can see that they accumulate more 4-HNE in the spinal cord. In the peak of the disease, we can measure pain. So we look at the EGR, which is a marker for neuronal activation. And we also look at the neuroinflammation. So we look at the astrocytes and the microglia in the dorsal horn spinal cord. And the two star 2 mice, they had increased neuroinflammation. And when we gave ALDA1, we clearly see a decrease in astrocyte and microglia expression, and also a decrease in the EGR staining. Okay, so far we had some more evidences that aldehydes is also controlling chronic pain. We also did for the chronic constriction injury, we have very good results. We also did for alcohol-induced neuropathy. It's a beautiful model, and we clearly see that the impairment in ALD2 activity is also important in this condition. So we are confident that it's an important enzyme to control pain, right? And I also have studies on opioid tolerance and hyperalgesia. I think okay, my battery. Can you hear me? Yes? Okay. And we also have nice studies on opioid tolerance and hyperalgesia that I will have to talk to you in another opportunity. Or as soon as we publish this paper, you can also check it. And when we give out the one, we can control inflammatory pain, chronic pain, and opioid tolerance. Okay, so for the second part, let's talk a little bit about how important is the fusion and fission process for pain control. So the mitochondrial dynamics is so we know that mitochondria are a dynamic and heterogeneous network, right? And the process of fusion and fission, they are controlled by specialized proteins like OPA1 and MFN2 in neurons, also MFN1, but the majority of the studies with the neuronal dysfunction are made with MFN2, but we also have MFNU, which is really, really important for the mitochondrial fusion. And the fission is mainly controlled by a cytosolic protein called DRP1, right? And this process is physiological. It's important for maintaining the oxidative phosphorylation and the ATP supply. And a balance between fusion and fission is critical for the mitochondrial function. So, because neurons have a high metabolic demand, we asked if the mitochondrial dynamics are involved in neuropathic pain. And as I mentioned to you, that chemotherapy-induced neuropathy is also a very common disease. We picked, and also based on literature showing that mitochondria was an important organelle for this kind of pain, we picked the chemotherapy-induced neuropathy. And why we think this is important? Because there is a huge incidence of cancer, right, and around the world, and mainly breast and lung cancer. They are very common. And in Brazil, there is an increased incidence and increased mortality, and that's why we oh boy, this video too big, I think. Okay. And we estimate increasing number of cases by 2040, around 47% of increase in cancers. So we have a problem coming up. And paclitaxel, it's a taxane, is one of the most common chemotherapies to treat solid cancer. So it's the first line chemotherapy to treat breast, ovarian, and lung cancers. It's an antimicrotube agent, right? That prevents, that induces microtube stabilization and prevents the cancer replication. However, it leads an injury of the or disease in the somatosensory nervous system, and around 70% of the patients feel some kind of sensory abnormalities. And among all these symptoms, neuropathic pain, it's a very important symptom, is common, it's very common, and that's why we believe it's very important. And sometimes the patient stop treatment, the cancer is getting better, but they stop treatment because they started feeling so much pain. So new alternatives are needed to treat this kind of pain. And we hypothesize that because neurons have a high metabolic demand, and the mitochondrial dynamics is important for cell homeostasis, we decided to look at the mitochondria in the DRG. And we didn't find lots of studies looking at the DRG, so we gave 4 injections of paclitaxel, like mimicking 1 round of treatment in humans. And we can see that after treating with paclitaxel, there is a decrease in the pain threshold, which persists for around 35 days, and then it returns to the normal baseline after 42 days. So we look at day 7, that we called early onset. We look at 20 days, that we called chronic, and 40 days, we call it as remission. And when we take the mitochondria, we clearly see the mitochondria is more vacuolized. There are some evidences that it's more fragmented and so on. So what we did, we measured, so there is an increased vacuolization when in the chronic phase. And we also saw an increase in GRP1 expression, which is a protein involved in mitochondrial fission, okay, in fragmentation. Okay, so there is an impairment in the mitochondrial dynamics. So how is the function? So we treat the mice with paclitaxel. We remove the DRGs in day 25, whereas the peak, and then we did an assay for mitochondrial function. We measured the oxygen consumption, and we can see that the paclitaxel induces an impairment in oxygen consumption, suggesting that this mitochondria is dysfunctional. And remember, if the mitochondria is functional, probably there are increasing oxidative stress, and 4-HNE is painful. It's a consequence of the oxidative stress. So we measure the 4-HNE in those DRGs, and we see it's increased. So there are evidence. So with this, we can assume that in the peak of the neuropathic pain induced by the paclitaxel, this mitochondria is dysfunctioning and is producing more aldehydes. We did it for the DRGs, and Natalia looked at the axon. So she removed the sciatic nerve, and she did all these biochemical studies. So in summary, she observed increased diffusion and decreased diffusion in the early phase and remission. But in the peak, in the chronic phase, she see a decrease in diffusion and an increased fission. Okay, and this increased fission is followed by apoptosis, like BAX-BCL2 increase, is followed by 4-HNE accumulation, by an impairment in oxygen consumption, and activation of MAP kinases like ERK and JNK. When we look to the axon, we can see axonal degeneration by she measured the myelin sheet, and she also measured the MBP levels, the myelin basic protein, and it's decreased in the peak of disease. And just to conclude my talk, as a proof of concept, we use tissue-specific knockout mice. So we knock it down GRP1 in NAV 1.8 neurons. In other words, we knock out GRP1 mainly in sensory neurons. And with this, we're supposed to decrease the fission, right? So here in green, we have the wild type mice, and the tissue-specific knockout display decreased hypernociception when compared to the wild type mice. suggesting that somehow if you decrease the fission, you can benefit the you can develop a less neuropathic pain induced by this chemotherapy. So suggesting that the fission process is really important for the development of neuropathic pain. And because we really like the translational studies, right? So we use the small molecule that sits in the FIS1, the place that the protein that connects to GRP1 to induce fission. So in other words, this small molecule prevents fission. And when we treat these mice with SC9, we clearly see a decrease in the pain induced by paclitaxel, confirming our results showing that the fission is important for the development of pain. And we just, as proof of concept, we removed the DRGs from naive mice. We just tried to recapitulate what we saw in vivo, in ex vivo experiments, in cell culture experiments. So we took the DRGs and we incubate with paclitaxel, and we clearly see an impairment, a cytotoxic effect of these neurons. We see that the mitochondria is more fragmented in the DRGs, in the mouse DRGs. And we also have a human model for sensory neurons, sensory-like neurons. We basically differentiate human neurons in sensory neurons. And again, when we incubate with paclitaxel, we see an increase in substance P release. We see a hypersensitization to calcium influx, which suggests that the paclitaxel per se is able to induce pain-like behavior in cells isolated. And these neurons also have more fragmented mitochondria. And when we treat these cells with an inhibitor of GRP1, we can also prevent the cell death induced by paclitaxel. And this study has been published. So if you are interested in go deeper, you can just go. So in conclusion, I hope you are convinced that ALD2 is an important enzyme in detoxifying toxic aldehydes, and that molecules that prevent excessive fission is also important for preventing neuropathic pain. I think that's it, and thank you. I hope I didn't pass much.
No, that was great. We had a question in the chat from Caitlin, and she was wondering about the sex of the animals in your studies. We do a lot of work in female rats versus male rats, comparing the way that they develop pain at the U of A. And so it would be interesting to know if there's any significant differences in your models of pain.
Thanks for your question. So we did for all those studies that I showed today, I didn't use females. I just used males. So I use females for the alcoholic-induced neuropathy. I use females for opioid-induced tolerance, and I use females for the chronic constriction injury. For those studies, unfortunately, I didn't have space in the animal facility to use females, but I'm sure that my referee will ask the same question. Ingrid, formoterol has been shown to ameliorate neuropathic pain in models using packet cell and is not this mitochondrial metagenesis.
Yeah, Ingrid has actually published on formoterol and pain. which is pretty cool.
Yeah, thank you for your question. I definitely have to check it out, right?
One of the questions that I had for you was regarding environmental exposures to lipid peroxidation with ALDH2. I'm not super familiar with LDH2 and what's known about it, but a lot of the work that is happening at the U of A right now is around how chronically being exposed to intense heat conditions can cause all sorts of negative effects, and being exposed to endocrine disrupting chemicals can have effects. Are there any exposure effects like that that are known for LDH2?
There is no study as far as I know. What I know is from smoke cigarette, vapes, the e-cigarette as well, pollution. There are some works, but nobody look at ALD2. There are few groups that are interested in ALD2 as a pain control.
Is it
It's a great
basically just your lab in Stanford?
question. Yeah, basically.
Okay.
And my husband, but he's interested in heart. Thank you, Ingrid. That's the location of
And
site.
we have a question from Melissa. She's asking if the localization of cells experiencing mitochondrial fission directly results in pain in that specific area of the body, or if it's more of a system-wide effect.
Yeah, we don't really know, right? That's why we started like looking at the neurons separated. We start looking at this the sciatic nerve. But as far as we know, I think it's global. Because if we are right, the consequence is increased aldehydes. So it can be everywhere, right?
So I have a not as scientific question for you. Um, I'm wondering what it's like to work at the Butantan Institute. We are mostly all at a state school institution in the US, and we have core labs and all these facilities that we can hire to do a lot of things for us. And we have our animal facilities. What kind of capacity do you have at the Butantan lab? What do you have to do for yourself, and what is available from the institute?
Now we do have a huge capacity. We have very nice facilities with technicians hired for each equipment. So I think the only concern here is the reagents. So we have to import them to US or Europe. So it takes some time to arrive here. I think this is the most issue here, but we do have facilities like Stanford, like you have in Arizona, and animal facilities. We do cross. We breed our animals in our lab in a smaller facility, but we also have a huge animal facility that give not give the best word in English. Sorry, that supply with rodents and also to feed, right, the reptiles and so on. So we have to have a huge animal facility for all those, because Butantan is mostly an institute devoted to study reptiles, snakes and scorpions and so on. I'm like a fish out of the water over there. But in my lab, people also study pain induced by snake bites and scorpions and spiders and so on. So I don't know if I asked your question, but I respond to your question. But we do have a huge capacity, and we just need to improve the way we get the reagents.
Well, now I need to know about the venom research. Are you studying the same mechanisms of pain, but with venom-induced pain? Is it the same topic as the rest of your work?
um, we had one important work where we noticed in the past, like in the 90s or even before, that the rattlesnake venom, when the person is bite by the rattlesnake, is not painful at all. It kills, but it's not painful. And we found a peptide inside this venom that's analgesic. So it's crazy that nature is so smart, so the snake doesn't need that the mouse escape, right? So when the rattlesnake bites, they are so okay, they don't escape, and the snake can eat well and fast. So we have studies on this, and we also have studies and where we establish like a library of peptides and molecules isolated from venoms, and then we can test and we do screening for analgesics. And now we have a very interesting peptide that seems to be analgesic as well.
That's really cool. That's amazing. Does anyone else have any other questions for Dr. Zambelli?
I have a kind of off-topic question as well. So if someone were going to come to your institute, would they need to speak Portuguese, or do a lot of the people there speak English?
We always speak English.
Yay!
We are receiving tomorrow a student. She's from New York. She studies here in São Paulo in an international school, and she's spending some time in our lab. So everybody speaks English, or if they don't speak English well, they can try. So you don't need to speak Portuguese to work at Butantan.
Wonderful. Well, thank you so very much for your talk. I really enjoyed it. As Sophia mentioned briefly, the professor I'm most interested in working with is doing pain research actually in cannabinoid receptors and opioid receptors. And he's looking at like natural products that he finds in cannabis that reduce addiction reward. And so I really found your research fascinating as well. So thanks for sharing.
Thank you, Caroline.
Yeah, I think this was great. I am actually kind of mind boggled by how much overlap you have between the things that you're studying and the models you're using and what we have at the U of A. None of the people that I've invited so far to speak to the drug club, who were from outside of our pain center, had that much overlap. So that's really exciting. And it's really cool to hear about the facilities that you have and the capacity that you have. I've heard horror stories about other countries and ordering antibodies and how difficult things can be. And so it's nice to hear that you have a lot of the facilities already set up and available. And the Butantan Institute sounds like a pretty cool place that maybe some of us would be applying to go to.
Yeah, we will be happy to have you here if you wish.
Well, you might get stuck with me and Caroline, so heads up.
Sophia and I both traveling. So I lived in Mexico for 7 years. I speak Spanish, but not Portuguese. But one of my good friends is Brazilian, and he's always joking with me that I just need to go to Brazil and learn Portuguese. So not having to, but being able to would be a great excuse, and immersion's the best way.
And we every time we have postdoc fellowships available for people abroad. So if you look at the FAPESP, I will. There are lots of opportunities, and the fellowship is quite good to live in São Paulo. So if you wish to study abroad or have some experience around FAPESP is a good, like our NIH.
Well, that's awesome.
Yeah. Thank you for sharing.
Yeah, thank you. All right, well, I would like to say thank you to Dr. Zambelli for talking to us about her research, and thank you everyone for attending and asking your questions. And that was really interesting, so thank you.
Thank you. Bye-bye.
Bye.